Now, this dream of helping type 1 diabetics, it's personal to me, but it's one that everybody should understand because it opens up the way for lots of other dreams. Let's talk about type 1 diabetes so you can understand it. And you should probably write some stuff down. So, first of all, most mammals have an organ called the pancreas. And the cells in the pancreas have the job of producing a protein hormone called insulin. Insulin is made in the cells of the pancreas. It's then exported from those cells by exocytosis. And then that hormone moves through the bloodstream to all the other cells in the body. The job of the insulin hormone is to get all the other cells in the body to allow glucose to come in so that those cells can be fed. If insulin is not available, those cells will not allow their food inside, and those cells will die of starvation. So there's a lot on the line here. Once scientists figured out the importance of insulin, they looked at people with diabetes and said, hey, maybe we can keep them alive by giving them insulin from other species. You know, if the diabetics' pancreatic cells were destroyed and couldn't make insulin anymore, maybe we just use insulin from cows or horses or pigs or some other mammal that's fairly closely related to us. And that did work. Injecting insulin from other mammals kept humans with diabetes alive, sometimes for decades. But the problem was that insulin from a horse is not as appropriate for a human as human insulin. So there were undesired side effects. But then someone had the dream. Couldn't we get bacteria to make human insulin for us? For example, we already saw that this species of bacteria could get this other species of bacteria to make more Fimbriae, more of these Velcro-like extensions. And it did so by simply transferring a gene over. Couldn't humans do that somehow? So yeah, that's how it works. So let's say we wanted to make a whole bunch of human insulin outside the human body so that it could be injected into someone with diabetes. What you do is you get hold of a human cell that has the insulin gene in it, and that would be pretty much all human cells, but grabbing one from the pancreas would work best. And anyway, you could try to grow that cell in the lab, keep it alive, and then you could determine where in all that DNA is the gene for how to make insulin. So the genetic code for how to make insulin, let's say it's right here, highlighted in orange. Once we have that identified, we're partway there. Now, the other thing we can do is we can look at a species of bacterium that's easy to grow in the lab. Now, bacteria typically have at least one large ring-shaped chromosome, but they also have lots of little tiny miniature chromosomes, and these are called plasmids. Scientists figured out that we could take a gene from a human being, like the one for insulin, and we could stick it into one of these ring-shaped miniature chromosomes called plasmids. And what we would get is what's called a recombinant plasmid, one that recombines the DNA from the bacteria, shown in purple, with the DNA from the human, shown in orange. And then we could take that ring-shaped chromosome and stick it back into the bacterium. Now that bacterium is genetically modified. It's mostly bacterial genetically, but it's partly human, and it has instructions on how to make human insulin. Now, it uses amino acids and ATP and ribosomes to make proteins. So as long as the instructions for how to make proteins or the DNA is human, then it should be able to make human insulin. Now, here's the great thing about doing this with bacteria. Bacteria reproduce themselves really quickly, once every 20 minutes. So that means they can double their population once every 20 minutes. Now, if you think about it, how many 20-minute long generations are there in one day? Well, at 20 minutes per generation, that's 3 generations per hour times 24 hours per day. That's 72 generations. So since theoretically the bacteria can double their population size 72 times in a day, that means in one day we could go from 1 to 4.72 times 10 to the 21st bacteria. So if we can get one genetically modified one and then give it all the food and water and warmth and surface area it needs, then we can grow a gazillion bacteria that are all genetically modified because they are all copies of their genetically modified ancestor. Now, it's really hard to actually have space and surface area for this many bacteria in a lab, so this is just a theoretical amount. But the point is, you get a ginormous army. They all make insulin. We have different techniques for collecting the insulin from the petri dish, purifying it, bottling it, and then selling it to diabetics. So the diabetics then have much longer, higher quality lives. And in the meantime, the company that figures out how to do this makes a lot of money. That is the genetic modification dream. And this occurred 40 years ago. It's as old as I am. 1977 was the year I was born.